Routes of Influenza in Mallard, February 7th, 2021, by Zach Harris. The title of this article was Alternate Routes of Influenza A Virus Infection in Mallard. The date the article was written was October 29th, 2018. The journal that the article was published from was Veterinary Research. The article was made by Michelle Will from the WHO Collaborating Center for Reference and Research on Influenza, Caroline Broher from the Department of Pathology and Wildlife Diseases, Ake Lundqvist, Zoonosis Science Center, and the Department of Medical Biochemistry and Microbiology, and Joseph Jarhold, the Section for Infectious Diseases, Department of Medical Sciences, and the last three were all from Uppsala University, which is where the Veterinary Research Institute is located. The acknowledgments listed is the Swedish Research Council, Formas, which funded the experiment. Introduction. The reason this experiment was conducted is because the starting place of influenza A, or IAV, is in wild birds. The wild bird that the researchers decided to focus on was the dabbling duck, or mallard. The research question for this experiment was, rather than just an oral or fecal route, can dabbling ducks contract influenza from cloacal drinking and or preening? Their hypothesis was that transmission of influenza or general disease is multifactorial in dabbling ducks, which includes oral, fecal, cloacal drinking, and preening. Methods. To start the experiment off, the ducks used in the sample were taken from a domestic owner and transported to the National Veterinary Institute in Sweden. They were then checked for influenza so they could be tested negative and decontaminated to start the experiment off clean. The ducks were then placed in 5 rabbit cages with holes underneath them to prevent infection. For the preening trial, where preening is just when the duck cleans its feathers with its beak, they were placed in a room so they could interact with each other. As for the virus stock was EID 50 and it was taken from a local observatory. Methods Part 2. For this experiment, the researchers had to conduct 2 sub-experiments in order to see if there were 2 more transmissions of influenza in mallard possible. The first one was cloaca drinking, and the second one was preening. The cloaca is essentially just the end of the digestive tract for birds. For this sub-experiment, 2 age groups were tested on to account for different digestive systems and internal structure of the mallard. The 2 age groups were 4 and 6 months. They were then injected by cloacal inoculation, which is just vaccination, and they were injected with 2 milliliters of virus stock using a blunt end metal cannula. The feathers were then sprayed with ethanol after 1 minute of vaccination to decontaminate their feathers. A separate group of 5-month-old ducks placed in individual boxes with collars on. Each box was filled with virus-laden water for 1 hour to inoculate them and so that they could perform cloacal drinking. The lights were turned off to relieve the stress of the duck, and in case anything bad happened, personnel were next to the duck just to make sure that they were relieved of stress. Immediately after this, the feathers were then sprayed with ethanol straight after. As for preening, which is just when the duck cleans its feathers with its bill, 10 6-month-old mallards were bathed in virus-laden water to get their feathers wet, while every other part of their body was decontaminated so that they could focus on the feathers. They were either 1 placed in cages, 2 placed in a room to move around in, or 3 placed with unaffected ducks as a contact to see how different forms of contact and infection could relate to each other. Methods Part 3. The ducks were sampled on 3 separate occasions: the start of the experiment, after 24 hours, and after 48 hours. The ducks were then euthanized so that the samples could be taken from them. Cloacal samples were taken by a sterile-tipped applicator and then stored at -80 degrees Celsius. Necropsies were taken after performing the euthanasia on all the ducks after 48 hours so that they could collect the gastrointestinal tract tissues. In the samples, if there was red staining in the nucleus or cytoplasm of the cell, this meant that the influenza virus replication was present. Results. For the cloacal drinking experiment, the cloacal swabs of all the birds came out positive, which should represent that influenza A had infected the duck. The viral replication showed positive stains for most of the birds, but not all of the birds. For the preening experiment, the gastrointestinal tract of every single bird showed staining and showed a positive viral replication. As you can see on the charts next to you, the preening shows a much more consistent infection with the cages, transmission of inoculation and contacts, while the cloacal drinking does show most of them being infected with influenza A with inoculation and cloacal. However, with inoculation in the 4 months, it was not showing as much infection rate spread as it had with the preening experiment. Discussion. Transmission efficiency plays a big role in influenza success for dabbling ducks. The reason dabbling ducks were used for the experiment is because the natural reservoir of influenza A is in wild birds. Unlike poultry, influenza A is not as threatening in dabbling ducks as it is in the poultry. In fact, it takes about 7 days before the disease goes away completely. Throughout the experiment, it was showed birds could become infected through cloacal drinking when swimming in water that was containing the virus of influenza. Contaminations of feathers were causing rapid spread of the infection, which is why in the preening experiment, the virus was spreading throughout the birds like wildfire. The researchers concluded that the virus is either contained in the bill of the duck or absorbed by the gland-secreted oils in the feathers, so it sticks to them. This is why this is a reason for the rapid spread of influenza during preening. So, true to their hypothesis, influenza can have many routes that now include oral, fecal, preening, and cloacal drinking. Credibility. I do believe that this research article is credible because it came from the Veterinary Research Journal, which is an international peer-reviewed journal. The impact factor of the journal is 3.432, which is over 3. Another factor in credibility was that there was a large sample of ducks purchased from a domestic breeder and raised at the National Veterinary Institute in Uppsala, Sweden. The experiment was funded by the Swedish Research Council Formas, so they had official funding. And at the very bottom of the research article, 39 references are listed as extra information that they took for their research. As far as uncredibility, I could not find anything except maybe the shortness of the experiment, which was 48 hours for the ducks. And but other than that, I didn't see any bias during the research article. For the works cited and references I used, the first one being the research article, and then the ones below that being the pictures used in the presentation.